And classification, phylogeny, and organization. Phylogeny is the evolutionary relationships among species. We're going to talk about how that relates to classification and organization. Systematics or taxonomy, those words get used interchangeably. Sort of. Systematics is the study of kinds and diversity of organisms, as well as that relates to the evolutionary relationships among them. As a result of systematics, we get taxonomy, which is a taxon are a grouping of animals that share a particular set of characteristics. Like Aves is the order of birds. So those birds are grouped together because they have feathers, they have beaks. Those characteristics group those particular organisms together. Taxonomic categories are hierarchically arranged in that they are grouped from less specific to more specific, the least specific being the domain, the most specific being the species. Please be aware that there are things such as varieties and subspecies of organizations. Those are not specifically defined, which is why they're not listed here. So for Homo sapiens, you have listed domains, the kingdom, phylum, the chordata, those are those with the notochord. Class, mammalians, they belong within the primate order. Family, Hominidae. Genus, Homo, species, sapiens. So you see Homo sapiens grouped together as the species. And then we see the same thing over here for dogs. Nomenclature is our naming system. It is a binomial nomenclature, meaning that it's two names. So we name organisms by their genus and species name. The individuals who get to do those are those of the International Zoological Association. The actual, we would call it a book, except it's probably not an actual book. It's probably kept on a computer, as the International Code for Zoological Nomenclature. So for humans, it's Homo sapiens. Some cases, genus may be abbreviated if it's stated previously. In some cases now, we look at DNA because DNA is a strong indicator of relatedness. Like for a long time, there was a big argument about where to put pandas. Are they bears, or are they more closely related to raccoons, or do they deserve their own distinction? Bears are grouped with carnivores, and because pandas eat bamboo, that threw things off. Then they kind of also were closely related to raccoons. It turns out when we look at the DNA, they actually are much more closely related to the bears than was originally thought. So now they are just classified with the bears. We find DNA in a variety of locations. The nucleus of the cell and the mitochondria, and of course ribosomal RNA, which is used in creating proteins. There are studies that look for evolutionary relationships, some of them using ribosomal RNA. Evolutionary change is very slow. So it's relatively easy to track. Domains as a group, there are only 3 of them. The Eubacteria having, of course, the bacteria in it, Archaea having microbes in it, and then we have the eukaryotes, which are going to have everything else in them. And those are categorized in that group because they have compartmentalized cells. So here, if we look at the lineages of life, you'll notice that this group over here, the eukaryotes, are definitely the largest component. The reason that we use systematics is that we're trying to find and define monophyletic groups. That is, groups that are descended from a common ancestor. So we want to know the single ancestral species and all of its descendants. Kind of like genealogy, I suppose. But we sometimes end up with members having polyphyletic groups, those being resulting from insufficient knowledge, or paraphyletic those are resulting from insufficient information. So if we look at evolutionary groups, polyphyletic groups are coming from 2 different ancestors. Monophyletic groups are coming from 1 ancestor. Paraphyletic groups include some, but not all members of a lineage. So you have 3, 4, 5, and 6. You're omitting 1 and 2. And these are the result of insufficient knowledge. So the way evolutionary systematics works is there is a traditional approach. In this case, what you look for are homologies. Now remember, homologies are similarities, traits that can be found similar between multiple species. We spoke of the bones, similar bones in forearms across multiple vertebrates. And those can be tracked across and used in classification. And then we put that information into a tree and you can even then apply time to it if you have that information. So these individuals groups are looking at, this is looking at vertebrate phylogeny. This particular tree I don't think is the best tree to look at, but we're going to look at some others that are more clear. In cladistics, or phylogenetic systematics, the most recent homologies are the most useful. Why? Because they tend to show the most recent evolution and most likely where the most recent breakoffs from those homologies are going to be. So you're going to look at ancestral characteristics that are common to all members of a group because they're going to indicate common ancestry. So you group organisms according to common traits. And those individuals are probably coming from common ancestors. You take individuals that are the group that's related that are not included in a study group, those are considered to be outgroups. Those are going to help decide if a character is ancestral or more recently derived. So if you have some individuals that are related and have the character, then it may be more recently derived. You just have to look at it and evaluate. Derived characteristics are those that have arisen since the common ancestry. They're also called synapomorphies. It's the same thing. Once you have this information, you can create a cladogram, and it's much more easy to see when you put it in to something visual, at least in my mind. I want to look at it this way as well, but maybe it is easier to look at it here first. Okay, so each one of the numbers are species. Each one of the letters represent a trait. So the trait created a branching episode. Why? Because every organism after this carried this trait, but the organisms before that did not. So this is a derived characteristic. Now, what we have further, so these individuals carried characteristic D. The species here, number 5, did not. It didn't have B, it didn't have D, it didn't have F or E. C was carried by 2 and 1, but not by 4, 3, or 5. Okay, does that make sense? Now, I think I skipped clade. Yep. Clades are related subset within a lineage. So, this would be a clade. This would be a clade. This would not be a clade if you chopped off one. That's not a clade. Just number 2 is a clade. Just number 3 is a clade. The whole thing is a clade. So if you include all related individuals, it's a clade. Now, phylogenetic species concept, all that says is that populations have evolved independent of other groups of populations. That doesn't mean that a population is not impacted by its environment. It just says that a population can't force another population to evolve. It's a result, it's not a cause. And that those individuals are, they may share derived characteristics if they're being affected by the same environmental forces. Hierarchical nesting means that you can have cladograms clades within a clade. That's what nested hierarchies are. So if you look, we go back to here for a minute. If this is a clade, then this is a clade within a clade. So all you're looking at are clades within clades. So if you can circle a clade within another clade, it's a nested hierarchy. Now, I can't really show you this well here, but if you blow it up, because it's going to go outside of my screen that's being recorded, but if you blow it up, what you're going to see is rather than numbers and letters are actual names of species and traits. So it's going to show the same thing, it's just actually going to give you real world examples. Okay. Symmetry describes how our animals are arranged around a pointer axis. I want to just show you what each of these is. Asymmetry, that means that there's no central axis or point, so it's not balanced. Bilateral symmetry, it means that's what you and I are. So arrangement of body parts around the single plane. You can cut it in half, and then the right and the left are going to be mirror images. Radial symmetry means that if you go around the central point, then you'll find structures in pairs or combinations around that central axis. So now to give you examples, sponges are, that particular one is asymmetrical. That coral polyp is radially symmetrical, and this fox is bilaterally symmetrical. So it's slicing through the plane. You can have two halves that mirror each other. Okay, other patterns of organization. The important things to point out here is that you can have unicellular organisms like protists and sponges at some points in their organization, in their life. More commonly, you can have diploblastic organization, which means that there's an ectoderm, an endoderm, and then there's a mesoglia, which is actually non-cellular. Okay? This explains those in more detail, which you can look up. There's also triploblastic organization. This is, we'll talk about organisms from each of these groups when we move on, but I'm just going to show you the terms for now. You can have acoelomates, pseudocoelomates, and coelomates. Coelomates are by far the most common, and it means that the body cavity is completely surrounded by a mesoderm. This is by far the most common. Pseudocoelomates you're talking about, and acoelomates you're talking mostly about. Pseudocoelomates and acoelomates you're talking mostly about some worms. Okay. Going back to taxonomy and relating it to symmetry, you can look this over, but it'll come more as we go on through the semester.